I posted the one from yesterday already. So, number one, make sure you know the equation for photosynthesis. Make sure you know that the reactants, carbon dioxide, which the plants get in how?
Leaves.
Keep going. Stomata, which on most plants are where? Bottom of the leaves. And why are they on the bottom and not on the top? Yeah, so that the water doesn't evaporate out as much, because water's coming out at the same time CO2 is going in. So it's about slowing down the rate of, what do we call that process that moves the water through the plant? Transpiration. But we said if a plant is like a lily pad or something, the holes have to be where? On the top. Not a problem because they're water plants. They need water, they get that through their roots. They make carbohydrate, which we might write as what empirical formula? CH2O, or we might just write it as C6H12O6, glucose. And oxygen we always have to write out, O2. And remember that this oxygen over here started out where over there? Oxygen over here, don't know what's going on with my marker, started out over here as water, okay, not carbon dioxide. That's terrible. Because during photosynthesis, what do we know happens to water? What happens to water during photosynthesis? It gets split. It gets split to replace electrons lost by what? Photosystem number 2. So we'll come back to that in a minute. Remember that part 1 of photosynthesis is the light-dependent part. Has to happen first. Has to happen in the light. And it happens where specifically?
In the leaves.
Leaves, zoom on in. Chloroplast, zoom on in. Zoom on in. The membranes of the thylakoids, which are these little sacs inside the chloroplast. That's where part 1 happens. Make sure you know that. And it happens there because what's embedded in the membranes of the thylakoids? Yeah, all the proteins that make photosynthesis happen, which are chlorophylls and keratins and cytochromes and all kinds of other proteins. That's mostly what we talked about last time, so let's do that real quick. So here's a picture of a single thylakoid. What's the area outside that thylakoid called? Stroma, with an R. But notice that there's all these things in blue are proteins. And they're stuck in the membrane. They're embedded in it. Why do they need to be stuck in a membrane? Why aren't they just floating around? Yeah, they got to be in a specific order, and it's tough to keep them in that order. So remember first, we've got this group of pigments, 2 big groups of pigments. And those are the photosystems. And they're groups of like chlorophylls mostly, but also some other colored pigments. And I told you to think of them like an array of what? Solar panels. Their job is to capture light, and then the energy that they capture, they're supposed to send that energy to a molecule in the middle, a molecule of chlorophyll A called what? What's the molecule in the middle of each photosystem called? Reaction center. And those things gain energy and gain energy and gain energy and gain energy until eventually they gain so much that what happens? They lose electrons. And what's the word for that? Losing electrons. Oxidation. When they lose electrons, they also lose energy. Electrons is a good way to transfer energy. And they usually lose electrons in multiples of what? 2. Because what's the metal that's part of chlorophyll? It's magnesium. And it's in a group of 2. So it loses 2 at a time. The electrons that leave photosystem 2 go across these proteins here between 1 and 2 called the cytochromes. We also call those proteins what else? ETC. They transport electrons. So the ones from 2 are used to do what to 1? The electrons from photosystem 2 do what to photosystem 1? They reduce it. The ones that left 1, the electrons that leave photosystem 1, end up with the help of this enzyme, NADP reductase. They end up reducing NADP plus to make NADPH. And I told you to think of that like charging up a rechargeable battery, charging it with the sunlight. So this is a charged up form of chemical energy, NADPH. It's an electron carrier, electron shuttle, electron storage device, something like that. NADPH stores energy that used to be in the sun. In the meantime, photosystem 2 lost electrons, it needs them back. Where does it get them? It rips apart water. We call that photolysis. It steals electrons from water, and what's left behind of the water are H pluses, protons, and oxygen gas. So the oxygen gas that you're breathing came to be, put it that way, when water was ripped apart by plants and algae and bacteria and such. So we start building up some protons in the middle here, the lumen or the space of the thylakoid. Another thing that builds up this gradient in here is that as these electrons are going across the ETC, their energy is used to power what? Active transport of protons from where to where? Yeah, what's the word? What's the word for outside? The stroma into the middle of the thylakoid, the lumen, the thylakoid space, building up this gradient. And I've compared that over and over again to stretching a rubber band, because even though they're getting pumped in, they want to go where? They want to go out because there's a gradient. And there's less of them on the outside than there is on the inside. So they want to go out, so it's a way to build up what kind of energy? Potential. Eventually, they can get out, but normally not, because this membrane is a phospholipid, and it doesn't like charged things, right? So they're stuck. But the way out is through this guy, which is ATP synthase. It's what kind of protein? Well, it's an enzyme, but it's also a channel protein, which means it lets what process happen through it? What kind of diffusion? Facilitated diffusion. And when it opens up, those H pluses shoot through it really fast. And this enzyme part of the protein can use the energy to put ADP and P together with some energy to make ATP. We call that what kind of process? What's the name for it? Yeah, but it's called chemiosmosis. Using energy from moving protons, proton gradient, to make ATP. So 3 things mostly made in the light part of photosynthesis, ATP and NADPH. Both forms of stored what? Chemical energy that came from the sun originally. And then oxygen gas. Another word we could throw at this picture to describe it is what? Another phrase. There you go. Non-cyclic photophosphorylation. Because electrons are moving from water to photosystem 2, across the ETC to photosystem 1, ultimately ending up in NADPH. They're moving, but not in a circle. And it's photophosphorylation because light energy is being used to phosphorylate what? ADP to make ATP. That's the light part of photosynthesis. We said the other part is necessary because NADPH and ATP are both what? Unstable. Can't transport them, can't store them. Most all this is happening in the leaves. So the plants have to have a way to get energy to the roots and to the stems. So in part 2, the Calvin cycle, they're going to use NADPH and ATP to make what? Sugar, which is more storable, more transportable, and so on. So why don't you try focusing here for a minute? We talked over and over again about the kinds of light the plants absorb best. And we know they absorb what the best? Blue, and then red, and they're very bad at absorbing green. Told you some numbers to know. Red is around 700 nanometers long, that's the wavelength, and blue's around 400. Know those numbers. Those are the colors that plants are best at absorbing. They're not good at absorbing 5 to 600, which is the greens and the yellows. And that's why they're green, right? That's what do they do to green? They reflect it, and what we see is what gets reflected. Mostly today, though, we want to focus on the Calvin cycle. Eventually, we'll focus on the Calvin cycle. So we looked at this picture briefly yesterday. Let's hit it hard today and talk about it. First of all, where does the Calvin cycle take place? Stroma, the fluid-filled part of the chloroplast. When does it take place? After the light dependent, but when is that? It's usually during the day. It's actually during the light, most of the time. Because what have we said most plants do at night? Most plants at night shut their stomates and sort of take a break. What's the term for most of the plants on Earth? C3. And that's what we're going to focus on completely here for a few minutes. And then we'll switch over. C3 plants are most of the plants around here. C4 and CAM plants are more plants you would find where? Real dry places, deserts, really dry places. So, in a C3 plant, right after the light-dependent reactions, you get the Calvin cycle. Calvin cycle starts with carbon dioxide coming into the plant. And in C3 plants, it most of the time goes into the middle of the leaf. What's the word for the middle of the leaf? Mesophyll. And that's where the most important enzyme for this is located at. It's called rubisco. Rubisco's job is to do what process? Fix carbon fixation. And that means take a CO2 and attach it to a carbon chain that's already in the plant. And that already present carbon chain is RUBP, ribulose bisphosphate. It's 5 carbons long. 1 CO2 attached to 1 RUBP gives you a 6 carbon chain. We normally think about this though happening 3 at a time. 3 carbon dioxides attached to 3 RUBPs. That makes an intermediate. And we said several times intermediates are things that are made in the middle of what kind of process? Metabolic pathway. It's something you have to make to get to where you're trying to go. It's not what you're trying to make at the end, but it's something you have to make to get there. This intermediate is short-lived, which means it breaks up. And these 3 6-carbon things break up into 6 3-carbon things. These 6 3-carbon molecules that we make, we usually call them PGA, the scientific name is phosphoglycerate. But I told you to think of these sort of like half of what? Half of a glucose, but what's it missing? It's missing the energy. It's like diet sugar. So the second part of the Calvin cycle is called reduction. And the second part function is to take that PGA and charge it up, add energy to it. Energy that came from where? The sun, and directly from where? NADPH and ATP that came from where? During the light-dependent part. So this ATP right here, these 6 ATPs were made during the light-dependent part. These 6 NADPHs were made during the light-dependent part. So what's one word could we use to describe what's happening to phosphoglycerate right in this part, right in here?
Phosphorylated.
It's getting phosphorylated. And we know what are the enzymes called that do that? Kinases. So there's some kinases that are taking phosphate from ATP, transferring it to PGA, charging it up with energy. And then down here, one word could we use to describe what's happening to the sugar? It's getting reduced because the NADPH is getting oxidized. NADPH is oxidized, the sugar is getting reduced. Both of these circled processes are putting energy into the sugar, energy that came from part 1 of photosynthesis. So we end up at the end of reduction with 6 molecules of this stuff called G3P, glyceraldehyde 3-phosphate. That is essentially half of glucose, but this time it has what? It has energy, lots of it. We make 6 of them, the plants make 6 of them, but they only take 1 out. That one that they take out of the cycle could be used to make glucose or fructose or starch or cellulose or whatever sugar a plant might need. The other 5, though, are used to carry out step 3. And step 3 is called regeneration of the carbon dioxide acceptor, or sometimes it's called regeneration of RUBP. If a plant runs out of this ribulose bisphosphate stuff, it can't work. It can't do photosynthesis. So, sounds wasteful, but 5 out of the 6 G3Ps are just used to make more of that, and that keeps the cycle going. That's the way photosynthesis happens in C3 plants. Light dependent, intelligent. That works great as long as there's plenty of what?
Carbon dioxide.
Plenty of carbon dioxide and plenty of water in the ground, right? As long as the ground is wet and the plant's got plenty of water and plenty of carbon dioxide, this works great. When it doesn't work great is when things start getting dry. And we'll talk about that here in a second. So this is where we left off yesterday. This is over here on the left is a leaf of how it looks in a C3 plant. What do we call the layer on top? Cuticle. It's really waxy, and it's sort of like what part of you?
Skin.
Sort of like your skin. Its job is to keep germs out, viruses, fungi. There's all kinds of things that will come after plants, and to keep water in, keep it from desiccating. And then we said on the bottom you got the stomates. What are these little green dots on each side of the stomate called? They're called guard cells. And their job is to guard the opening. They can either open it wider or they can close it up. And then in the middle, the mesophyll, all this area between about right here and right here is mesophyll. This is where the cells that do photosynthesis are at. And we said the ones down at the bottom are all spaced out. For what reason? Why is there lots of open space down there?
There's a lot of carbon dioxide to get in.
Yeah, the CO2 is all coming in through here. So it has to make its way up to the top. So there has to be air spaces for that to happen. That's why those are there. And then also inside the mesophyll are these veins. And inside those veins, that's where what 2 sets of pipes are. Xylem and the phloem. And we said yesterday that the xylem carries mostly water, and it carries it mostly in what direction? Up, from root toward leaf. And the process that carries it through there is called transpiration. Phloem is the other kind of tube, and it's mostly carrying sugars, and it's mostly carrying it which way? Down, from leaf to stem to root. Anybody know the process that moves stuff through the phloem? No? It's called translocation. And we will talk about that once we finish up evolution. That's one of the few things we need to cover with plants. But we'll get there. Now that's a C3. Notice a C4 leaf looks kind of different. In a C4 leaf, the bundle sheath cells are way thicker, and also they're surrounded by the mesophyll, sort of compartmentalized. And might as well talk about that now. So some C3 plants that are out there are legumes. What are legumes? Bean family. Peanuts, beans in general. What ability to do a lot of those legume plants have that makes them pretty important to nature? They do nitrogen fixation. Take nitrogen and convert it from N2 to what? Ammonia, or what other ion? Nitrate. It's either ammonium ion or nitrate. But it also includes wheat, oats, barley, rice. So most of our crop plants, C3s. Like I said, they work great as long as there's plenty of water in the ground, plenty of carbon dioxide in the leaf. But that becomes a problem when it starts getting hot. When it gets hot and the ground gets dry, they're forced to do what? Why does that happen? What do they have to do physically? If it's really hot and really dry, what do the plants have to do physically? Close their stomata. When they close their stomata, what happens to the concentration of CO2 in the leaf? It goes way down. And what happens to the concentration of oxygen in the leaf? It goes up. And we said that RUBP is not all that great of an enzyme because it's equally attracted to what? Oxygen and carbon dioxide. Normally not a problem because how much oxygen is there normally in a leaf? not too awfully much. But when the stomates are closed, there's a huge amount. So instead of connecting CO2 to the carbon chain, it connects oxygen to the carbon chain. And that's called photorespiration. Because does that lead to anything good? No. Doesn't lead to sugar. It leads to like toxic waste products that eventually the plant is going to have to undo. It's going to have to use energy to undo. So that's bad. There you go. And if photorespiration happens for a long enough continuous stretch, what happens to the C3 plant? It either dies or goes what? It goes dormant. Like your grass turning brown in August, right? It's not dead, but it's dormant. So that limits C3 plants' abilities to live in really, really hot places. C4 plants are better at living in hot places. C4 plants are things like sugarcane, corn, Bermuda grass, which a lot of you probably have in your yard, sorghum, which is the stuff we use to make molasses out of. They're a little bit better adapted at dry things. And one of the reasons they're better adapted is they have an additional enzyme. It's called PEP carboxylase. And we said that PEP carboxylase in a C4 plant is found out here in the mesophyll. Stomates are down here. Carbon dioxide comes in. PEP carboxylase fixes carbon. It does sort of what Rubisco does. Fixes the carbon. Fixes it in the mesophyll. But once it catches that CO2, what does it then? What do C4 plants then do with it? They take the CO2 from the mesophyll and they transport it into these little bundle sheath cells. And what did I compare the little bundle sheath cells to, since they're a small enclosed area? When we talked this through first time, I compared it to like a closet. And what's locked up in the closet? No. Well, the xylem and phloem are in the red part, but I'm talking about in this area around the red part. This is where C4 plants keep Rubisco locked up. If he's locked up, he can't get in too much trouble, right? So the PEP carboxylase in a C4 plant, they fix the carbon out here. They then transport it into this littler space, the smaller space. Rubisco's in there. They turn it loose, the CO2 loose. Rubisco will then carry out the Calvin cycle. So they fix the carbon initially in the mesophyll, move it to a different place, and then do the Calvin cycle. We say those two processes are separated temporally. But that works. That's important, because what does that let them do? What can they do that C3 plants can't do? They can close their stomates for long periods of time, because PEP carboxylase has no attraction for what? Oxygen. And even if there's just a little bit of CO2 out here in the mesophyll, PEP carboxylase will grab it, transport it into this little space. And even if there's just a little CO2, if it's in a really small space, but still high, concentration. So Rubisco still works, it still does its thing. So C4 plants open and close their stomates off and on all day and still do photosynthesis. That's good. That's not the best version of this, though. The best version are cam plants. Cam plants are mostly what kind of plants? Cactuses, also pineapples, desert plants in general. Now they have PEP carboxylase just like C4 plants, so that's one adaptation. But what do they do that most plants don't? Yeah. They open their stomates at night. And all night long, they're breathing in carbon dioxide. PEP carboxylase is fixing that carbon dioxide, and it's storing it as an acid, a 4-carbon acid. Usually it's called malic acid. To store it up. Storing carbon dioxide all they can, all night long, building it up. As soon as the sun comes up, what happens? Close the stomates, because what happens in the desert when the sun's up?
It's hot.
It gets really hot really fast. But at night it's cool. So when the sun comes up, then that acid they've been storing all night breaks back down and releases what? Carbon dioxide. So there's a constant supply of carbon dioxide, and what enzyme then takes over? Rubisco, and they do Calvin cycle. So all plants do Calvin cycle. C3 plants go light dependent straight to Calvin cycle. C4 plants do light dependent, then they fix carbon with carboxylates, transport it to the bundle sheath, and then they do the Calvin cycle. CAM plants fix carbon at night, store it up all night, and then they do the Calvin cycle during the day. Go ahead.
Have you ever done a lab where you control when the stone is open and closed in microscope so you can see it?
I haven't, but one thing that we're going to do sometimes in the next couple of weeks is you can take clear fingernail polish and rub a coat of it on the bottom of a leaf. And then you can peel it off and put that on a microscope slide, and guess what you can see? You paint the bottom of a leaf with fingernail polish, pull that off after it dries, and put it on a microscope slide.
Yeah.
It's like fingerprinting a leaf. Okay, but you don't have fingerprints, but you can see the imprints of what? Stomates. And you see some plants have lots and lots of stomates, some not so many. What kinds of plants would have lots and lots of stomates? Plants that live in what kind of places? Like a rainforest, where you don't have to worry about what? We don't have to worry about water. Plants that live in drier places are going to have fewer stomates because transpiration and water loss is a problem. We'll look at that as we go. All right, one other thing that's not directly related to photosynthesis, but sort of is, that we'll review here. This pyramid is like a food chain, right? And we're going to call it a trophic pyramid. What does trophic mean? Like heterotroph, autotroph, feeding or eating. So it's a food pyramid essentially. What do we know we always call the things on the bottom of one of these pyramids? They're the producers. And most of the time we call them the primary producers. Those are the things that do photosynthesis. They're going to be plants or cyanobacteria or some kind of algae. And then this level, here they call them herbivorous consumers. But what do most things call them? Primary consumers. They're the plant eaters, right? And these we usually call the secondary consumers because they eat the primary ones. What's after secondary? Tertiary. What's after tertiary? Quaternary. And I don't know what you call it, probably quint. Fifth level, something like that. But this is a pyramid for several reasons. It gets smaller as you go up. One reason it gets smaller as you go up is what goes down as you move up. The amount of energy that's available. We'll talk about that in a minute. What else goes down as you go up? The number of organisms, or maybe the mass of the organisms, the biomass, but normally it's the number. The reason the energy goes down, because what kind of energy are these producers using? They're using the sun. There's lots of energy available to them, right? Some of that energy they'll use and give off as what? Heat. A lot of times they're releasing heat. Also, when whatever these things are, clams or whatever they are, eat this phytoplankton, some of the energy that was in these cells, they can't use, right? They can't get it out. So it just like comes out in the waste. What percent of the energy that's stored here makes it and is used by these? About 10%. Call it the rule of 10. So if these things stored up 1,000 calories, only about 100 calories make it to here and are used, right? And only about 10 calories make it to here and are used. And so on. The rule of 10. So the amount of energy goes down by how much as you go from one level to the next? It goes down by 90%, right? Only 10% is transferred, which means it goes down by 90% as you go from one level to the next. Does that work? Here we can kind of see that. This one doesn't start with a producer. They probably need another level down here with some kind of producer, but here we got shrimp being eaten by bleak, which I assume are some kind of little bitty fish. The bleak are eaten by perch, which are bigger. Those are eaten by pike, which are even bigger. What's an osprey? A bird. It's kind of like a water version of an eagle, right? A smaller version of an eagle. All right, we're not going to deal with that right now. let's switch over and we'll talk about respiration for a little while. And then we'll call it quits. All right, so not going to get too deep into this today. Hit it hard in the morning. First of all, what organisms on Earth do cellular respiration? Everyone. Plants, bacteria, fungus, it doesn't matter. Most do what kind of respiration? Aerobic. Some do anaerobic. Actually, most do what? Most do both. But they mostly do which one? Aerobic. Like you. You're usually doing aerobic respiration. Occasionally, you have to do some anaerobic. And when would that occasion be? Yeah, when you're really stressing your body, you're sprinting, you're lifting heavy stuff, and you can't get enough what? You can't get enough oxygen to your muscles, so you have to do something else to make energy, and that something else is what we call fermentation, or anaerobic respiration. Make sure you know the equation for aerobic respiration. It's just exactly the same as what? This is cellular respiration. It's exactly the same as photosynthesis, just flipped around backwards. So for aerobic respiration, we need some glucose. We need some oxygen. Those are the reactants. We also need some ADP and some P. And what we're going to do is essentially do what to the glucose? What did we say respiration really is? What kind of reaction? It's a combustion reaction. You're burning it, slowly chemically burning it. And whenever you burn a carbon-containing thing, you always make three things: carbon dioxide, water vapor, and some form of energy. It might be heat, it might be light. In this case, it's ATP. You release those forms of energy. So again, know the equation, know the reactants, know the products, know how to balance it, know that the reason for doing this is to make the ATP. Because what does ATP do for you? It provides what? Free energy, activation energy, to make pretty much everything in your body go. So it's hugely important. All right, this is probably about as far as we'll get on this slide. So what came first, aerobic or anaerobic respiration, according to scientists? Anaerobic. Anaerobic. Because what were the first things, living things on Earth? Bacteria. And what hadn't started yet? Photosynthesis. So they're fairly certain that there was no oxygen in the beginning. There were other gases, like maybe methane, maybe some carbon dioxide, maybe some nitrogen. So the first things couldn't have been aerobic because there wasn't any O2. There was oxygen, but where was most of the oxygen at? Bonded to what? Most of it was bonded to hydrogen in the form of water, right? Photosynthesis broke down that water and released some of that oxygen, and then aerobic respiration evolved later. But the first things were anaerobic, probably bacteria, didn't use oxygen. And oxygen is actually toxic to some things. It's actually really reactive chemical, right? Yep. Another important thing is today, cell respiration works basically the same in all known organisms, whether it's a bacteria, a fungus, a plant, a human. Cell respiration is an example of a what? It's a metabolic pathway. And we know that each step in a metabolic pathway requires different what? Well, they make different intermediates, but require different enzymes. Enzymes are made out of what? Proteins. So if we do the same reactions, we have the same enzymes, what else must be pretty much the same? Because the DNA is very similar, right? So that's where we're going, is that this is an example of what we call common descent or unity that all these things share these pathways according to scientists because they share genes, because they share what? Common ancestors. They're related somewhere down the way. We're going to end today by mentioning briefly the first step in aerobic respiration. Aerobic respiration is simplified down to 3 steps. Glycolysis, step 1. Where in a cell does glycolysis happen? Cytoplasm. Or sometimes, what else do they call cytoplasm? Cytosol. And step 2 is the Krebs cycle, which is also known as citric acid cycle. And it happens where in the mitochondria? The matrix, the innermost compartment. And then the electron transport chain, which we also sometimes call oxidative what? Phosphorylation, happens on the little inner membrane of the mitochondria, and those little folds on there are called cristae. Finally, maybe, glycolysis. I want to go to a diagram, and we'll pick up on this tomorrow. Here's the metabolic pathway of glycolysis. To start glycolysis, first of all, what has to happen to the glucose? What are we doing in that first step? Phosphorylating it, which means we're adding what? Energy. But what are we trying to do? Break it down to do what? To get out energy. But the first part of glycolysis is sometimes called the energy investment part, because you have to put in energy in order to get out energy. So we phosphorylate the glucose, we phosphorylate it again. That makes it what? Unstable. So we're trying to lyse the glucose, break it. So you make it unstable, you break it apart, and we'll go from there. But the last thing I'll mention today, what's the enzyme called that does that second phosphorylation step? Phosphofructokinase. And we talked about it over and over again because what does it have on it that's really important? It has an allosteric site, feedback site. And what binds to that allosteric site? End product of cell respiration, which is what? ATP. And it's a way to do what with this whole process of respiration? Negative feedback, turn it off until you need to turn it back on, right? It's a way to temporarily turn it off. That is a good place to stop.